and may not be knowing that how RT-PCR works. So that we will discuss over here. So if we see the polymerase chain reaction, we know that it is nothing but where if we have the DNA, so it's something like that xeroxing of the things, that is a Xerox machine. So PCR is nothing but it's a technique. It's not a machine, it's a technique that is called as polymerase chain reaction, which is done on thermal cycler. So what exactly is done here is the DNA is been copied from one to the several millions of the copies in few hours. So it's called as polymerase chain reaction. So this method can generate tens to billions of the copies of a particular sequence of interest. So whenever the polymerase chain reaction or PCR is carried out, it is carried out by keeping the particular gene into a target. So target identification we can see. So basic principle of PCR lies that it serves to replicate and amplify the target DNA, prepare master mix which contains the DNA templates, DNA polymerase, nucleotide that is called as DNTPs, the primers. Then it is programmed in a 3-step cycle which are repeated from 20 to 30, and sometimes it is also repeated up to 45 cycles, but not more than that, because all the techniques has its own limitations. So the PCR process proceeds in 3 distinct steps governed by the temperature. So the first step is the double stranded DNA. All we know that is only be amplified by PCR technique. So the double stranded DNA is denatured by the heating typically at 95 degrees C to separate the double strand, and that is called as denaturation. That is done near about 95 degrees C. Thereafter, the second step is the reaction is rapidly cooled to an annealing temperature to allow the oligonucleotide primers to hybridize to the template so that the DNA polymerase which are used can amplify onto the template DNA of the template DNA. So the annealing temperature vary from 50 to 65 degrees C depending upon the system in which the PCR is used. Either it is prokaryotic or eukaryotic. Usually the prokaryotic annealing temperature is a little bit lesser, near about 55 or something like that. But while the eukaryotic annealing temperature is a little bit at a higher side, it's nearly it is beyond the 65 degrees C. So thereafter, the third reaction is called as extension, which is done at 72 degrees C. The reaction is heated to the temperature typically at 72. I'm sorry, it is 72 degrees C for the efficient DNA synthesis by the thermostable DNA polymerase. And as in thermostable DNA polymerase, there are the constituent the DNA region which is to be amplified. So that is called as target DNA or the template DNA. Then the primers. There are 2 types of primers which are used, which is called as forward primers and the reverse primers. So that are complementary to the 5 end of the each of the sense or the antisense strand of the target DNA. So is the primer, because the primer has 3 end OH group, and that OH group allows the DNA polymerase to add up the nucleotides. So that is called as polymerization steps are being carried out by the tag polymerase usually, but for more efficiently PFU polymerase, that is called which is isolated from the Pyrococcus furiosus, is also used, which has a greater efficiency than the tag polymerase. The tag polymerase is an enzyme which is extracted or isolated from a thermophilic bacteria, Thermophilus aquaticus, and hence it is known as tag polymerase. So it has a high fidelity of the DNA polymerization in vitro and in vivo also. So the deoxynucleotide triphosphate DNTP, which includes that DATP, DCTP, DZTP, and DTTP, that is deoxy. So it can have the opportunity to add up by the DNA polymerase to the primers, then to that of the 1-by-1 addition of the nucleotide onto the template DNA. Then the buffer solution, that is the 1X buffer, is usually used to provide a suitable chemical conditions for the optimum activity and stability of the DNA polymerizations, that is the PCR reactions. Then the divalent cations, which are required cofactors for the DNA polymerase, that is Taq polymerase, and the PFU polymerase. So any divalent cations like Mg2 plus or Mn2 plus can be used to as a cofactor for the Taq polymerase enzyme. Now, these steps can be initial denaturation, so it is done only for once. Then the step 1, 2, and 3 is repeated in a cyclic manner, that is 20 or 30 or 40 or something like that. So 1 and 3 steps are repeated. Initial 2 minutes, 10 minutes, or 5 minutes, initial denaturation is given, and final extension is also given. So initial denaturation and the final extension is not been included into the cycle, as the cycle starts from step 1, but initial denaturation is the first step of the initiation of the PCR reaction, and the final step is the final extension, and thereafter it is put on hold. So this is the reaction cycle of the PCR. This is very basic one, and think now. So what happens? The DNA during PCR, the DNA which is used as a template is first denatured in first step, then and second step the annealing allows the primers to bind. So there are two primers, forward and reverse primer. So both primers binds onto an opposite strand of the DNA. Then in extension, DNA polymerase is there, which starts adding up the nucleotides onto the primer, as the primer 3 end site has OH group. So the RNA polymerase can only adds up the bases on 3' and only. So the progress of the DNA synthesis in PCR occurs from 5:3' end. So 5:3' end, and after second to third cycle, the original amplification is started, and the amplification can continues up to the steps or the cycles we keep in PCR. But the product which is made or formed in PCR has its own limitations, and hence we usually do not keep the cycle more than 45 for the small primers, and the usually the PCR reaction is carried out up to 30 to 35 cycles as per the requirement. So what is the limitations of the traditional PCR, and why do we have RT-PCR in our use or for our use? Is it has a low sensitivity, poor precision, results are not expressed as numbers, as it can have in a appendop tube, that is PCR tube, which can only be seen with the ethidium bromide, means electrophoresis putty gel stained with ethidium bromide, or the particular staining procedure, either or silver staining, which is not a quantitative, it is just a qualitative, that how much DNA is present, and the amount of DNA which are generated or amplified into the product can be a crude method to be estimated when we run the PCR. So post PCR processing is required, and therefore somewhere you might have heard of, you might have seen, that is the gel documentation system, which allows us to some extent to read that what is the band size and all, but it is just a crude method. So quantification or quantitative analysis is not usually feasible accurately with the PCR product. So it has a narrow dynamic range with less than 2 logs. Now the alternative method of the PCR to do the quantification analysis is the northern blots, RNA protection assay, in situ hybridization, competitive PCR, cDNA array, and all. But we don't have any kind of graph observed or obtained by using this traditional PCR. And apart from that, the additional or the alternative methods which we do have in our hand has an additional problem associated with it, and those problems are like difficulty in achieving the high throughput, then using large RNA or DNA quantities, limited dynamic range, threat of contaminations, difficulty designing controls, and difficulty of creating and optimizing the quantitative assays. So goals to improve this problem is to eliminate the use of electrophoresis, gel electrophoresis, which is usually carried out after the PCR reaction, To increase the after increasing the reproducibility, only we can have the problem to be sorted out. To enable us to internal control on a standard, reduce turnout time, increases throughput, and reduce the sample amount uses will definitely be the goal for to improvement in the basic PCR or the alternative method. So hence the quantitative PCR has emerged or led to us to overcome all these problems associated with either PCR or the alternative method which are used in place of the PCR. So detection of PCR product growth if you use the quantitative PCR, it collects the data during high precision exponential phase, and even in a small amount of the DNA, we can have a large, enormous data to be produced by the quantitative PCR or real-time PCR. If we see that the real-time PCR, quantitative PCR, actually these are the types of PCR which are usually been taught and is in applications. So there are like touchdown PCR, colony PCR, hot start PCR, and in that RT-PCR has double meaning. One is called as real-time PCR, which is also called as qPCR, and another is the reverse transcriptase PCR, so reverse transcription PCR. So when we see or say the real-time PCR, it is nothing but a qPCR. So the real-time PCR has an or requires an instrument which consists of three main components. One is thermal cycler, then optical module to detect the run, and then a computer which can control the thermal cycler and to extrapolate read the data in terms of the graph, and hence the gel documentation system can be or use of the gel documentation system can be eliminated. So how the real time PCR looks is, this is looking like a general PCR, but it is normal PCR, but this real time PCR has a component where either this LED array light will fall onto that of the sample through the top or through the bottom. There are different types of PCR machine, and hence the software and the hardware